Welcome to episode 3 of the BioLogic podcast. Today I'm going to be discussing proteins, a school of organic compounds unrivaled in its variety and utility. Your body utilizes nearly 100,000 different proteins. That's 100,000 different combinations of amino acids within a polypeptide chain. Proteins are a polypeptide chain of amino acid subunits. The chain folds itself into a particular conformation depending on the amino acid subunits that it contains. Sometimes this protein, this peptide chain folded into a particular shape, is able to provide a useful function. Sometimes the protein performs a function that is harmful to the host organism. This is the case for prions, or prions, if you will, a class of misfolded proteins that can distort and misfold other proteins, leading to neurodegenerative diseases like mad cow disease. These protein chains, called polypeptide chains, are made up of amino acids. There are 20 different types of amino acids, and they can be arranged in an enormous number of ways to wield an equivalently enormous number of possible proteins. Each of these amino acids has a basic structure that consists of a carbon molecule bound to an amino group on one side, a hydrogen atom in the middle, and a carboxyl group on the other side. These two groups have an ionized and non-ionized form that they continually switch back and forth between. The non-ionized form is the neutral molecule, with hydrogen bound to the oxygen in the carboxyl group, and two hydrogen atoms bound to the nitrogen atom in the amino group. In the ionized form, the carboxyl group loses that hydrogen atom. The loose oxygen atom becomes negatively charged. Meanwhile, the nitrogen in the amino group attracts another proton, giving itself a positive charge from this additional hydrogen atom. Ultimately, what this means is that the core structure of the amino acid molecule is polarized in its ionized state, positive on the amino end and negative on the carboxyl end. This quality is involved in the linkage of these peptide units into a polypeptide chain, but I'll get to that in a few minutes. You might recall that I only mentioned 3 bonds with the central carbon atom, and you know that carbon can bind with 4 atoms, so what's the deal with the 4th bond? The 4th valence electron of the carbon atom is shared with a side chain. This side chain, called an R group, is the structure that distinguishes the amino acid and gives it a particular polarity and function. There are 20 different R groups. 10 of them are nonpolar, 5 of them are polar, 2 are acidic, and 3 are basic. The nonpolar amino acids are all hydrophobic, preferring to interact with other nonpolar molecules while avoiding contact with water molecules. You'll see the importance of this trait when we get to the lipid bilayer membranes of the cell. The 10 nonpolar amino acids, their short names, letter symbols, and their side groups are as follows. Notice the lack of strongly electronegative atoms in these nonpolar amino acids. Glycine, side group Gly, symbol G, with an R group consisting of a single hydrogen atom. Alanine, short name Ala, symbol A. It has an R group with a single carbon atom. It's bound to 3 hydrogen atoms. This is also called a methyl group. Valine, short name Val, symbol V. It has an isopropyl R group, 1 carbon atom with 2 more carbon atoms attached to it. Leucine, short name Lu, symbol L, with an isobutyl R group. It's a 2-carbon chain with 2 more carbon atoms attached to the last carbon in a branching formation. So 1 carbon, 2nd carbon, and then 2 more branching off. Isoleucine, short name Isle, symbol I. So it's got a 4-carbon R group with 3 carbon atoms in a chain, and the 4th carbon atom is bound to the 1st carbon atom directly attached to the amino acid. So isoleucine is shaped like if you hold out your thumb, just your thumb and forefinger like an L. That's pretty much what isoleucine is shaped like. It's a little chunk of carbon shaped like that. Methionine, short name Met, symbol M. It's an interesting R group. It consists of a 4-atom chain. The first 2 atoms are carbon, followed by a sulfur atom, and then another carbon atom. Cysteine, short name Cis, symbol C. It's a 2-atom R group consisting of a carbon atom bonded to a sulfur atom. Phenylalanine, short name Phe with PH, symbol is F. Its R group consists of a carbon atom and a cyclohexene ring, a hexagonal 6-carbon ring with alternating double and single bonds between the carbons in the ring. Tryptophan, short name TRP or TERP, a symbol W. It has a complex R group consisting of a carbon atom and then a pentagonal ring consisting of 4 carbon atoms and 1 nitrogen atom. The 2 most distant carbon atoms of the pentagonal ring are part of another cyclohexene ring shared with 4 additional carbon atoms. Proline, short name Pro, symbol P. It has an R group of 3 carbons, and these 3 carbons are in a chain. So interestingly, the 3rd carbon on this chain loops around and binds to the nitrogen atom in the amino group, taking the place of one of the traditional hydrogen atoms. So the polar side chains all contain 1 or more atoms with a particularly strong electronegativity. This imparts a polarity, which makes, that segment of the protein hydrophilic. The 5 amino acids with polar side chains are as follows. Serine, short name Ser, or SER, symbol S. It's an R group with a chain consisting of a carbon atom followed by a hydroxyl group. The oxygen in the hydroxyl group has a partial negative charge, while the carbon has a partial positive charge. Threonine, short name TRH or Thr, symbol T. The R group is an isopropyl structure, except one of the branching carbons is replaced by a hydroxyl group. So again, there's that oxygen atom with the partial negative charge. Tyrosine, short name TYR or Tyr, symbol Y. It's an R group chain consisting of a carbon atom bound to a cyclohexene ring with a hydroxyl group on the far end of the ring. So again, that oxygen atom provides the polarity. Asparagine, short name ASN or ASN, symbol N, with an R group chain of 2 carbons. The last carbon atom is a ketone, having a double bond with an oxygen atom, connected to an amino functional group. Glutamine, short name GLN, symbol Q, with an R group chain the same as asparagine, except with 3 carbons in the initial chain instead of just 2. The following 5 side chains are electrically charged. Some are acidic, some are basic. These side chains, they don't have the modest partial charges seen on the hydroxyl group on many of the polar side chains. Instead, these electrically charged side chains have a full positive or negative charge on their atoms, none of this partial charge silliness. So a negative charge means that one side chain is acidic, as it has donated a proton. A positive charge means the side chain is basic, as it has accepted a proton. A negative charge means the side chain is acidic, as it has donated a proton. The proton is positively charged, hydrogen nucleus. It's positively charged, and so when it leaves a molecule, it takes that positive charge with it, and there's a negative charge then left on the molecule. And so a positive charge, on the other hand, means that the side chain is basic, as it has accepted a proton. And so by accepting a proton, the molecule takes on that extra positive charge. The 2 acidic side chains are aspartate, short name ASP or ASP, symbol D, with an R group consisting of a 2-carbon chain, the last carbon of which is a carboxylic acid, or a carbon with a double bond to an oxygen and a single bond to a negatively charged oxygen that lacks a proton. So if you hold your thumb, forefinger, and middle finger straight out in an L shape, your middle finger and your index finger will be like the double bond to the oxygen atom. Your thumb, on the other hand, will be like the single bond to the oxygen atom with a negative charge. This is the structure of the carboxylic acid on the end of the R group of aspartate. Glutamate, short name GLU or GLU, symbol E, with an R group similar to aspartate, except it's got 3 carbons in the primary chain instead of just 2. The 3 basic side chains are lysine, short name Lys or LYS, symbol K, with a 5-atom R group chain consisting of 4 carbon atoms capped on the end with a positively charged nitrogen atom. So this is a nitrogen atom with 3 hydrogen atoms bound to it. That 3rd hydrogen atom brings in that extra positive charge that takes it from equilibrium into a positively charged state. Arginine, short name ARG or ARG, symbol R, with an R group consisting of 3 carbon atoms in a chain, followed by a nitrogen atom, and then another carbon atom bound to 2 amino functional groups, one with a single bond and one with a double bond. When in an ionized state, the extra proton can be on either nitrogen. Histidine, short name His, symbol H, with a stubby R group consisting of a carbon atom attached to a 5-atom ring. Two atoms in this ring, the second and fourth, are nitrogen atoms. The positive charge that comes from an extra proton can be added to either nitrogen atom. So that's it. All the protons are folded chains composed of a random assortment of these 20 amino acids. So a quick recap of what I just covered. The amino acids are composed of a base carbon atom, attached to an amino group, a carboxyl group, and a specific side chain. These amino acids connect together like links in a chain. While in an ionic state, the negatively charged oxygen in the carboxyl group on amino acid A takes two hydrogen atoms from the positively charged nitrogen atom in the amino group on amino acid B. By taking these two hydrogen atoms, the oxygen breaks its bond with the carboxyl group. The oxygen and its two hydrogen atoms break off and become a molecule of water. While that whole process is going on, the nitrogen atom in amino acid B and the carbon atom in the carboxyl group of amino acid A bind to each other in the process of water formation, which is called a condensation reaction. A condensation reaction involves the production of a water molecule. This creates a peptide bond, a single bond between a carbon atom and a nitrogen atom with nearly the strength of a double bond. The valence electrons on the nitrogen atom are shared with the electrons in the peptide bond, which stabilizes and strengthens the bond as a whole. When any group of subunits bind together to form a chain or fiber structure, the subunits are called monomers, and the resulting structure is called a polymer. The act of monomers binding to or joining a polymer chain is called polymerization. Polymerization is a condensation reaction, or a dehydration reaction, which produces a molecule of water. When a polymer is broken down into individual subunits, this happens because of a hydrolysis reaction. These names and their functions are easy to remember. You should know what condensation is. Water in the atmosphere collects on a surface and coalesces into drops of water. You see it on your windows, on your glass mug, on your body in a steam room. Similarly, the condensation reaction creates a water molecule. If you want to call the reaction by its other name, a dehydration reaction, think of it as a reaction that takes a water molecule from two binding monomers. The binding monomers have thus been dehydrated. As for hydrolysis reactions, on the other hand, the root word hydro means water, and lysis means to break apart. We'll see the root terms lysis a lot more when studying the immune system and physiology. The purposeful destruction of specific cells is often carried out by inducing lysis, or destruction of the membrane and collapse of the cell. The hydrolysis reaction involves the consumption of one molecule of water to split the bond between two monomers. The atomic components of the water molecule get incorporated into the molecular structure of the monomers, which is precisely the opposite of the condensation reaction. Spontaneous polymerization of amino acids in a solution doesn't happen as often as spontaneous hydrolysis, which makes it rare for long polymer chains to form or last for any appreciable length of time. Experiments that have attempted to recreate primordial conditions on Earth have shown that long polymer chains can be created and sustained in specific environments. This includes the sulfur-rich gas ejected into the cold ocean water by undersea vents, within undersea volcanoes, and on a variety of mineral substrates. Anyways, however it happened 4 billion years ago in the Archean, these polymers persisted long enough to undergo chemical evolution and help create the first self-reproducing molecular structures and the first cells. These amino acid polymers are called peptides, or oligopeptides or polypeptides, depending on whether or not they have less or more than 50 amino acids. Fewer, they're called oligopeptides. More than 50 monomers, and they're called polypeptides. When peptide chains are produced in a solution, they appear as a residue of organic matter. When peptide chains are produced in an experiment, for example, the resulting products are called amino acid residues. Whatever you want to call them, they're chains of amino acids, and they have 3 important characteristics that determine how they fold and function. These 3 characteristics are flexibility, directionality, and a shared orientation among their R groups. So the valence pair on the nitrogen atom of the amino group inhibits rotation at the carbon-nitrogen peptide bond, although this doesn't stop the rest of the single bonds in the polypeptide chain from bending and rotating, so this gives them a degree of flexibility. The polypeptide chain also has an amino group end, called the N-terminus, and a carboxyl group end, called the C-terminus, due to the repeating nature of the monomers. New monomers are added to the N-terminus of the polypeptide chain. This induces a directionality into the chain. As a result of the directionality of the amino acid monomers, all of the R-group side chains are on the same side of the polymer. Most fascinatingly, this gives the R groups the ability to interact with one another depending on their polarities and structure. So what is this folding that I keep mentioning? What does it mean to say that an amino acid polymer needs to fold before it can become functional? I'll go into more detail in a moment, but it means that the chain bends and wraps it around itself in a specific pattern which depends on the structure of the chain itself. So there are 4 general levels of structural organization that exist in a protein, or a folded peptide chain. The first level of structural organization is the primary structure, which I haven't named yet but have already discussed. The primary structure is simply the unique sequence of amino acids in the chain. Because there are 20 different amino acids, for any given polypeptide chain of length n, there are 20 to the nth power possible sequences of amino acids. There are over 10 trillion possible amino acid combinations for a polypeptide chain that's only 10 monomers long. Think about that for a moment. The number of mathematically possible polypeptide chains is incomprehensibly huge, and that's just for a short, stubby 10 monomer chain. There are peptide chains that are dozens and dozens of monomers long. The mathematical limits for protein variety are virtually endless, and yet the human body only uses about 100,000 different kinds. Think about all the proteins that don't exist naturally, but could be made in a lab. Think about how many of them might produce a protein or an enzyme with amazing functions, or functions that no one ever conceived of. If it isn't obvious, this suggests an absolutely tremendous opportunity for discovery. This is a whole new frontier of science. If you want to study proteins and the functions of amino acid chains, you need to look up a field of science called proteomics. Anyways, let's get back on topic. The primary structure of the amino acid is very important. It influences not only the initial 3-dimensional conformation of the chain, it influences every subsequent level of structural organization. The second level of this organization is called, surprise, surprise, the secondary structure. The secondary structure is the result of hydrogen bonding between the carbon-oxygen groups and the hydrogen-nitrogen groups in the backbone. Because a lot of hydrogen bonds are produced in the secondary structure, the entire 3-dimensional structure of the protein is greatly reinforced and stabilized. This hydrogen bonding among the backbone monomers contorts sections of the peptide into an alpha helix or a beta pleated sheet. In an alpha helix, hydrogen bonds form between monomers that are 4 positions apart in the primary sequence, twisting the backbone into a coil or helix. In a beta sheet, the chain folds back on itself, and hydrogen bonds form between adjacent backbone monomers. The secondary structure that happens to emerge depends on the charges and geometry of the amino acids in that part of the polypeptide chain. Consider the amino acid proline. Proline is a nonpolar amino acid, and its 3-carbon chain reaches around to bind with the nitrogen in its amino group. Proline can be incorporated into beta sheets, but will terminate in alpha helix because its specific geometry distorts the helix shape. The tertiary sequence of the polypeptide is determined by the actions and interactions of the side chains. Once the polypeptide is folded into its alpha helixes and beta sheets, the interactions of the side chains create the tertiary structure. When the side chains interact with each other, or other parts of the backbone, these interactions will twist and bend the polypeptide. Every minor twist and bend contributes to the total 3-dimensional shape of the protein. These side chains interact with each other, or the backbone, in 5 different ways. Some I've already covered, some I haven't. The methods of interaction that the side chains engage in are hydrogen bonding, covalent bonding, ionic bonding, van der Waals interactions, and hydrophobic interactions. Hydrogen bonds form between the polarized atoms in the R groups and the backbone or other R groups. Covalent bonds appear in the form of disulfide bonds, bonds formed between the sulfur atoms and the sulfhydryl groups on the amino acid cysteine. Ionic bonds exist between the acidic and basic bases, as well as other atoms in a side chain that can receive a full charge, not just a partial charge. Van der Waals interactions occur between hydrophobic or nonpolar molecules. So the constant motion of electrons, in their electron cloud, induces patches of very small polarity differences along the molecular structure. When nonpolar molecules get really close together, the van der Waals interactions become strong enough to influence the structure and orientation of the polypeptide chain. Hydrophobic interactions also occur between hydrophobic and nonpolar molecules in a water solution. The polar water molecules interact with polar R groups, while nonpolar hydrophobic R groups are forced to clump together. In these clumps, they form more hydrogen bonds, and it makes them like tough little knots in the polypeptide chain. The primary, secondary, and tertiary structures are levels of organization that exist for individual peptides. The fourth level of structure, quaternary structure, involves the combination of two or more peptides to form a complete multi-part protein. Quaternary structure combines peptide subunits with the same types of bonds that are used in the tertiary structure. Proteins that are composed of two subunits or two folded peptide chains are called dimers. 3 subunits makes a trimer, 4 subunits makes a tetramer, and so on. Hemoglobin, for example, is a tetramer. Hemoglobin is a protein that allows your blood cells to absorb and transport oxygen atoms. The 4 subunits of the hemoglobin tetramer include 2 copies of 2 different peptides, called alpha and beta chains. All of these structural levels ultimately depend on the primary structure of the polypeptide, the original amino acid sequence. The hydrogen bonds between amino acid bases make a secondary structure that is influenced by the physical geometry of the side groups. The interactions and geometries of the side groups determines the tertiary structure, and the quaternary structure depends on how the tertiary structures of 2 or more proteins can fit together. In order for any of this to work, the polypeptide chain has to be folded in the correct shape. It has to have the correct 3-dimensional shape in order to properly function, in order to properly fit into the structure of some larger protein, like a piece of a 3-dimensional jigsaw puzzle. If the polypeptide chain is folded improperly, or folded properly but then unfolded, or denatured, the resulting protein won't work. It won't fit where it needs to. Protein folding makes the polypeptide molecule more stable than it was as a free-floating chain, and it enables the creation of complexes of molecular machinery without which life is impossible. In the case of a protein denatured by some added chemical agent, the removal of that agent will cause the protein to return to its properly folded configuration. There are small molecules called chaperones, or heat shock proteins, which guide and encourage proper folding. They're called heat shock proteins because they're generated in large quantities after the cell is exposed to heat. Heat is basically energy in transit, and when the cell is heated, it's receiving energy. So depending on the temperature or the quantity of energy added to the system, the hydrogen bonds and eventually the covalent bonds will break. So when the cell is exposed to heat, the tertiary structure of its proteins may break apart and collapse. Heat shock proteins are thus produced en masse to facilitate the repair of a huge number of denatured proteins. I believe Dr. Rhonda Patrick has discussed heat shock proteins while being interviewed on the Joe Rogan Experience podcast, which is a great podcast. If you haven't heard about it, you should check it out. So most proteins, when in their final configuration, contain regions that are disorganized, a mess, unused for any particular reaction or interaction. Only small sections of the protein, called active sites, may actually be involved in any potential reactions. Furthermore, some proteins, they can't fold themselves into a functional shape without being bound to other ions or molecules. So when I say the protein is in a functional shape, I'm referring to its active state. The active state is the specific structure of the protein that allows it to react or perform a particular function. An active state implies an inactive state, which is the specific structure of the protein that makes it dormant and unable to function. The difference between these states can be as little as a single ion binding to the active site. During the transition between states, the protein structure will typically change. Consider hemoglobin. Each subunit has an iron molecule surrounded by 4 nitrogen atoms and a carbon ring, all in a single plane. When an oxygen molecule binds to one of the 4 iron atoms in the subunit protein, it induces a conformational change that twists the whole molecule, including the other 3 subunits. By causing this molecule-wide conformational change, the hemoglobin expresses a higher affinity for binding oxygen. Essentially, By binding one oxygen molecule, the hemoglobin becomes better able to bind other oxygen molecules. That's a particularly useful feature if you need molecular oxygen carried from your lungs to all your tissues. Another example of proteins changing shape between active and inactive states is calmodulin. Calmodulin is a signaling protein that regulates blood pressure. In its inactive state, the calmodulin molecule looks like two relaxed lobes, kind of like a pair of berries in the same stem. Each lobe is composed of 4 short alpha helixes, all clumped together. After you drink a glass of milk, or eat a ton of cheese, you're going to swamp your stomach with calcium, which then goes into your bloodstream in a big wave of calcium ions. Because the concentration of calcium ions in your cells and bloodstream rises, calmodulin begins to bind to these ions. The act of binding to the calcium ion, an electric particle with a double positive charge, causes the calmodulin molecule to transition to its active state. The transition involves the connecting alpha helixes from each lobe to extend until they meet and become a single long alpha helix bridge between 2 groups of 3 shorter helixes. 2 calcium ions can bind to spots in the folds of each group, making the activated calmodulin look like a dumbbell. Once in its active state, the calmodulin can send a signal to the cell to expand. When all the cells lining your bloodstream start to get the signal by the calmodulin molecules that float inside of them, they all start to expand. When all your cells expand, the blood vessels dilate, your blood pressure lowers. When the concentration of calcium in the blood is lowered through metabolic consumption, the calmodulin molecules bind to calcium ions less often. The signal to expand is sent less often, and fewer cells expand until the calcium levels in the blood return to normal. In this way, calmodulin regulates your blood pressure. These 2 examples demonstrate just 2 of the hundreds of the thousands of functions that proteins carry out. The versatility and adaptability of proteins is both unprecedented and unequaled in the chemical world. Without proteins, there's almost nothing in life that could function. Proteins do or are involved in virtually everything that the cell does to survive and reproduce. Proteins in the cell are used for structure, for transport, for defense, movement, signaling, and for catalysis. Structural proteins are often shaped like fibers and tubes. They wrap around the edges of the cell. They extend across the cell, and they act as scaffolding for other proteins and structures moving around in the cytoplasm. Transport proteins assist in the movement of a substance from one point to another. Some transport proteins carry ions throughout your body, like hemoglobin, while other proteins are built into the cellular membrane and regulate the passage of a particular ion or molecule into and out of the cell. Of the membrane-embedded transport proteins, some are like tubes that only allow a particular ion or molecule, like water, through. Others are like gates, bending to open only after they bind with a certain ion. Defensive proteins protect cells and tissues by attacking pathogens and other antibodies. The immune system is a hugely complex subsystem with its own patterns of rapid chemical evolution, almost entirely composed of proteins. Movement proteins are like chemical workhorses, and they're responsible for physically moving or carrying stuff. When your cells need to move large molecules, they use motor proteins. When you use your muscles to walk or stand upright or blink or anything, you're using motor proteins. Signaling proteins, like the calmodulin I discussed a few minutes ago, are responsible for relaying information within an individual cell and for relaying information between groups of cells. Enzymatic proteins are those which speed up or catalyze a reaction. Proteins that are capable of catalysis are called enzymes. Enzymes make reactions happen faster by lowering the activation energy. This is also the energy needed to initiate the reaction. Enzymes are really fascinating. By making it easier for a reaction to occur in the first place, the enzymes make the reactions occur with greater frequency. When the reactions occur more frequently, the macroscopic effect caused by the millions of reactions, like calmodulin dilating your blood vessels, happens faster and the effect is stronger. There are so many reactions and interactions that depend on enzymes to make them possible that without enzymes, life could not exist. I'll cover enzymes more in a future episode of the BioLogic podcast, but that's it for this episode. I hope you enjoyed it, and I hope you learned something cool about proteins. Next episode, episode 4, I'll be exploring another class of important biomolecules, the essential nucleic acids. If you enjoyed the podcast, tell your friends, tell your teachers, tell your classmates. hit the like button and subscribe so you can see new episodes when they release. The BioLogic podcast is kept alive by the support of listeners like you. If you want to help support the podcast, please consider buying some merchandise in our official store or becoming a patron by visiting our Patreon page. The official BioLogic store can be found at redbubble.com/people/biologic/shop. In the store, you can buy merchandise like t-shirts and coffee mugs and hoodies. All the products have really cool biology-themed artwork on them that I've designed myself. The official BioLogic Patreon page can be found at patreon.com/biologicpodcast. The support of Patreon patrons is very important for the show. Longtime patrons are rewarded with exclusive episodes on fascinating topics and will have their questions prioritized in AMA episodes. Thanks for your support, and as always, thank you for listening.